Hello viewers. Today we are going to start session on metabolism of amino acids and proteins. In this we are going to discuss about nitrogen balance, catabolism of amino acids, urea cycle and specialized products from amino acids and human chlorophyll. In these contents only selected topics are going to be covered. So let us begin with metabolism of nitrogen. Nitrogen, which is an important biomolecule compounds are obtained like amino acids and other nitrogen based substances. Hence, nitrogen plays a very important role and it is an important element along with carbon, hydrogen, and oxygen for living organisms. It is required for the formation of amino acids, proteins, and nucleic acids, and vitamins and hormones. So, these amino acids, proteins, nucleic acids, vitamins, and hormones contain nitrogen element. So let us see what is nitrogen fixation. Our atmosphere contains 78% nitrogen, but this nitrogen is not available for higher plants and animals. So this atmospheric nitrogen should be converted to useful form by a process called as nitrogen fixation. So to get the nitrogen present in the atmosphere for the living organisms, it has to be fixed. So let us see how this nitrogen fixation occurs. Let us see definition. Conversion of free atmospheric nitrogen into nitrogenous salt to make it available to the plants is called nitrogen fixation. Nitrogen fixation mainly carried out by nitrogen fixing bacteria like Azotobacter, Clostridium and Rhizobium, and these very helpful in fixing the nitrogen. Nitrogen fixation is carried out by enzyme system called nitrogenase complex. So this nitrogenous enzyme complex which is present in the above said microorganisms is helpful in nitrogen fixation, the crucial components of which are dinitrogenase reductase and dinitrogenase. Nitrogenase complex converts atmospheric nitrogen to biologically useful form, that is ammonia, which can be used directly by living organisms. So this is about nitrogen fixation where it is converted to ammonia form and later it is converted to various nitrogen based biomolecules. Let us see nitrogen assimilation. Ammonia which has been taken by plants are incorporated into a cell's biomolecule, mainly amino acids. This process is called as nitrogen assimilation. Nitrogen fixation, which is a atmospheric nitrogen, is converted to ammonia, that is called as nitrogen fixation. And the nitrogen assimilation means conversion of the ammonia into useful biomolecules such as amino acids and other compounds is called as nitrogen assimilation. Glutamate provides the entry point to ammonia assimilation. The glutamate is very important compound which can take ammonia. Glutamine synthetase, which catalyzes the formation of glutamine, is the key enzyme in the nitrogen assimilation. So let us see in the depiction. Glutamate plus ammonia, they both combine with energy and the energy is obtained from ATP, and this process takes place in presence of glutamine synthetase, and hence ammonia combine with glutamate, you will get glutamine, and subsequently the ATP which was there is converted to ADP. So hence by energy, ammonia in the reactant area combine with glutamate to form glutamine with release of ADP. In plants, glutamine gives glutamate by glutamate synthetase reaction. Glutamine plus alpha ketoglutarate in presence of glutamate synthase is converted to two molecules of glutamate by conversion of NADPH to NADP. So this is about conversion of glutamine to glutamate. Once ammonia has been fixed into glutamate, this ammonia amino group can be transferred to different amino acids to form used to produce different amino acids. These amino acids are used in the formation of proteins in the plants. Animals then use plants as source of amino acids to build their proteins and other nitrogen containing compounds. So nitrogen fixation for conversion of atmospheric nitrogen to ammonia and assimilation from ammonia to amino acids, and especially glutamate plays a very important role in binding the ammonia. And that glutamine, which is a amino group carrier, is helpful in the synthesis of other nitrogenous compounds and amino acids. When organisms die, microbial degradation of the proteins forms ammonia in the soil. So this is again comes in nitrogen cycle. Whenever the biomolecular nitrogen compounds they decompose, they will release ammonia into the atmosphere. Next is protein turnover. So the protein is a part of living organisms along with carbohydrate and lipids, vitamins and minerals. The body proteins are in a dynamic state. They are continuously renewed by degradation, that is catabolism, and resynthesized by anabolism. This process is called as protein turnover. Average protein turnover rate is around 400 grams per day. It's like you are earning money and you are spending money. So whatever the money that is earned is spent for various purposes. Similarly, protein turnover also occurs in the same way. Whatever the protein we will take, it is utilized by the body for various processes. So that is called as usage of protein and consumption of protein. So consumption of proteins is also be used in the body. Hence, it is called as protein turnover. Average protein turnover in a person is 400 grams per day. Amino acid pool. So what is amino acid pool? It represents the amount of free amino acids present in the body at any given time. It is about 100 grams. So free amino acids are available in our body which can be used for protein synthesis process. Sources of amino acids in the pool are amino acids are formed from breakdown of body proteins. By catabolism of proteins, we will get the amino acids. And second is amino acids that are synthesized in the body, that is biosynthesized in the body. Such type of amino acids, especially non-essential amino acids. And the third one, amino acids that are obtained from dietary proteins, that is from the food through nutrition or from diet. So these are the three reasons by which amino acids are present in the body. The fate of amino acid in the pool are the amino acids which are present in our body is used for first one production of proteins, second one is formation of specialized products, and third one is catabolism of amino acids to form keto acids and ammonia. So this is first one is protein turnover, second is about amino acid, how it is produced or present in the body and how why it is used for. Next is nitrogenous balance. Nitrogen, which is represented as N. The difference between intake and output of nitrogen compounds is known as nitrogen balance. How much nitrogen enters into the body and how much nitrogen exits from the body, that represents the nitrogen balance. Three nitrogen balance conditions are stated. So that is first one, you can see nitrogen equilibrium or nitrogen balance. Next one is negative nitrogen balance, and third one is positive nitrogen balance. So first one is nitrogen equilibrium, second one is negative nitrogen balance, and third one is positive nitrogen balance. So let us start with nitrogen equilibrium. A person is said to be in nitrogen equilibrium when the nitrogen intake, mainly in form of proteins in the diet, is equal to the total nitrogen output. So amount of nitrogen intake, you can see underlined in the blue color. The nitrogen intake left side is equal to the nitrogen output. If you take 100 grams of nitrogen containing the diet, and the output of nitrogen from the body in the various forms is same, about 100 grams, then it is called as nitrogen equilibrium. Normal healthy adults will be in nitrogen equilibrium. So especially in the adult age, the nitrogen balance is in equilibrium. So negative nitrogen balance, if the nitrogen intake is less than the nitrogen output, then the person is said to be in negative nitrogen balance. For example, if you take 100 grams of nitrogen containing diet and the output of the nitrogen from the body in the various forms is 120. That means extra 20 is going out, that is going to form negative nitrogen balance. So in the underlined red color you can see nitrogen intake and nitrogen output. So nitrogen output is more than the nitrogen intake. There is a net loss of protein nitrogen in the body. This condition is seen in starvation, protein energy malnutrition, wasting diseases like tuberculosis and illness. So negative nitrogen balance is going to affect the body, because since nitrogen is lost more in the body proteins which is present in the muscles and other deposits, that nitrogen is taken for maintaining the balance. Hence it leads to various type of protein malnutrition conditions or negative nitrogen balance symptoms. So hence it is the negative nitrogen balance I underlined in red color. The red indicates danger, hence it is color is selected to underline it. So negative nitrogen balance is not good for the body. So next is positive nitrogen balance. If the nitrogen intake is more than the nitrogen output, then the person is said to be the positive nitrogen balance. For example, if you take 100 grams of nitrogen compounds and the output of the nitrogen is only 80, that means you are taking 100 but output is only 80, so 20 remains in the body, that means you are in positive nitrogen balance. So 100 minus 80 plus 20, that is 20 is remaining in the body. There is net retention of the protein nitrogen in the body. This condition is seen in growth periods, especially in the children and pregnancy, lactation and convalescence after illness. Note that high intake of proteins does not lead to positive nitrogen balance. It increases both anabolism and catabolism of proteins so that the nitrogen equilibrium is maintained. So it is underlined in green color to indicate it's a good condition, positive nitrogen balance, where nitrogen is retained in the body, building the body. Next, we are going to discuss about amino acid related reactions. We will begin with the transamination. Trans means transfer, amine means amino group transfer. So amino acids, you know, as the name indicates, amino means NH2, acid means COH group. So NH2 group, amino, COH group, acid group related reactions can be seen in the amino acid reactions topic. Let us begin with transamination. Transamination is a process of transfer of amino group from an amino acid to a keto acid, converting the original amino acid to a new keto acid and the original amino acid into a new keto acid. That means converting keto acid to new amino acid and amino acid to a new keto acid. So without liberation of ammonia. So let us make it very simple what this paragraph means. So transamination is the process of transfer of amino group. That's the first line. So in the depiction, if you see the before reaction or arrow mark, that is reactant, and after the arrow mark is a product. So before the arrow mark we can see amino acids which contain amino group, and plus keto acid too, that is a keto acid is also available. So the amino acid which is having NH2 group, amino acid 1, it is going to donate NH2 group to keto acid 2. By loss of NH2 group from amino acid 1, you can follow the blue color line, it will get converted to keto acid by loss of amino group. From amino acid 1 will become keto acid 1, that is keto acid 1 is a new keto acid, that is mentioned in the second line. You can see converting the original keto acid. You can see in the first line, transfer of amino group from a amino acid to a keto acid, converting the amino acid to a keto acid, and then thus keto acid 2 before arrow mark, by getting the amino group, it is converted to follow the red line into amino acid 2. So keto acid 1 is obtained from loss of NH2 group from amino acid 1, that is following the blue color. Amino acid 2 is obtained from the addition of amino group to keto acid 2 followed by red line. This takes place with the help of transaminase reaction in presence of a coenzyme called as pyridoxal phosphate. You can see in the second paragraph, transamination reactions are catalyzed by transaminases. Reaction is transamination. Obviously the enzyme is transaminases, or it is also called as aminotransferases. Pyridoxal phosphate is the coenzyme for this or these enzymes transaminases. This is about transamination. Next one, the actual example for transamination. If you see aspartate, that is amino acid 1, and aspartate, that is amino acid keto acid 2, that represent the whatever I was shown here, the depiction, that is general depiction, that specific example is shown in this slide. The aspartate will become to a oxaloacetate, which is a keto acid, and aspartate, by getting the amino group from aspartate, is followed by orange color converted to glutamate. So aspartate by loss of NH2, blue line following to a keto acid, that is oxaloacetate. Aspartate, which is a keto acid, by taking the amino group following the orange color, you will get a glutamate, that is a amino acid. This reaction takes place in presence of aspartate transaminase AST in presence of a coenzyme pyridoxal phosphate. Similarly, you can see one more example, alanine transaminase, which is also called as SGPT. Alanine plus alpha ketoglutarate. So it is converted to pyruvate and glutamate. Alanine, which is a amino group containing compound, that is going to lose NH2 group, converted to pyruvate, and NH2 group is obtained by alpha ketoglutarate, is converted to glutamate by this process. Next is oxidative deamination. Just we have discussed transamination. Now we are going to talk about deamination. In transamination, there is a transfer of amino group from one compound to another compound. That means from amino acid to keto acid. In deamination, only removal of amino group, not transferring, only removal. The removal of an amino group from glutamate to release NH3 and alpha ketoglutarate is catalyzed by glutamate dehydrogenase enzyme. This exclusively takes place in liver. If you see the reaction, general reaction, as I told you, removal of amino group. Here I have shown glutamate plus water. In presence of glutamate dehydrogenase, NAD is converted to NADH plus H plus. Alpha ketoglutarate is obtained by loss of NH2. Glutamate is converted to alpha ketoglutarate. That NH3 is going to combine with one of the molecules of hydrogen to form NH4, that is ammonia. Hence this ammonia is obtained in the free form. Hence this is called as deamination. In this you cannot see any keto acid compound. Transamination over, deamination over. Next one is decarboxylation. Since in this you can see amino acid means containing COH group and NH2 group, amino and acid group. When amino acids are decarboxylated, corresponding amines are formed. This reaction is catalyzed by carboxylase enzyme, which require PLP as coenzyme. So in this depiction, you can see NH2CCOH. That means this example or general representation of amino acid above R and H. So different types of amino acids having basic amino group and a carboxyl group. It undergo decarboxylation in presence of enzyme decarboxylase. Arrow mark, you can see above the arrow mark, the enzyme is present decarboxylase. And as delete first two letter DE, delete means remove. Hence below the arrow mark, you can see removal of carbon dioxide. Hence it is called as decarboxylation. Obviously the name is related to the process happening, and it takes place in presence of enzyme PLP. By loss of CO2, you can see the comparison before the reaction and after the reaction. In the after the reaction, you cannot see one carbon and two oxygen. You will get amine, only NH2 is present, NH2CH2R. Hence amines are obtained from the decarboxylation process with the help of enzyme decarboxylase. This is our decarboxylation reaction. Next one is formation of histamine. This is general representation decarboxylation. Specific example I am going to show you. This is also decarboxylation. Histidine gives histamine on decarboxylation by enzyme histidine decarboxylase enzyme. Hence histidine arrow mark histamine. Above the arrow mark decarboxylase enzyme. From which compound the decarboxylation is taking place? From histidine. Hence the enzyme name is histidine decarboxylase. And below the arrow mark, obviously since decarboxylation, you can see CO2 is released in the presence of coenzyme PLP. This histamine product, that is after the arrow mark, you can see is a brain producing substance. Hence you can see histamine is a powerful vasodilator. It reduces the blood pressure. Histamine regulates the HCl secretion by gastric mucosa. It's also participate in allergic and inflammatory reactions. So this is about the decarboxylation reaction. We'll continue the next session. Thank you very much.